All right, so like Michelle said, my name is Brock Hedges, and I'm looking at the conservation and management of Australia's southern freshwater granite rock holes. And I'm using environmental DNA to sort of address some questions around invertebrate communities in them. So I'd like to begin today by acknowledging the traditional custodians of the land on which I've undertaken the majority of my fieldwork research. So those are the Cookatha, Wirangu and Barngarla peoples. And I'd like to also acknowledge the Kaurna people of the Adelaide Plains whose land I'm presenting from now, and acknowledge elders past, present and emerging, and extend that acknowledgement to anyone that is tuning in today. All right, so the rock holes that are the focus of my research form in large granite outcrops that sort of burst out of the surface of the surrounding soil, and they're present across much of southern Australia. They fill up following rainfall and provide a quite unique habitat. So invertebrates hatch out of eggs that have been stored in sediments, and some of these sediments have been baked at really, really hot summer temperatures. They're also a plant community that similarly sort of comes out of seeds that have been stored in these sediments as well. But why are these rock holes important, and why is it interesting to understand how they are used? So firstly, they're important for the conservation of a bunch of native species of conservation interest. So things like yellow-footed rock wallabies that are observed to be in decline across Australia. These animals move long distances through the semi-arid and arid regions of Australia to find sources of fresh water. So any sort of reliable surface fresh water can be really important to their conservation and management. They're also being used by a variety of pest species. So things like goats and rabbits and cats have all shown up during my period researching these rock holes. So understanding how these animals are using the rock holes as well can also be really important to their management. And they're also used by a series of less understood Australian species. So they provide a bit of an opportunity to better witness these animals and try and understand their life cycles. And finally, as they're rain-fed and entirely weather-dependent, we need to increase our understanding of sites like this to improve our response to climate change and climate change-associated threats, for which the rock holes are expected to experience a significant degree of. So the rock holes I work on are out at Hilltop Nature Reserve, which is here in South Australia. So it's about an 8 hours drive northwest of Adelaide. But I've also visited other rock holes in granite across Australia, and I was hoping to visit a few more, but with COVID and the pandemic, that's gotten a bit harder over the last 2 years. So this is what Hilltop looks like. I've been really lucky to be able to access it and work in collaboration with the Nature Foundation, who manages it. And this is some drone photography that we took on one of my recent trips out there this year. So you can see that the landscape is sort of completely populated by these granite outcrops and these rock holes present on them. So the rock holes have been well known for thousands of years and have actually been traditionally managed by many of Australia's Aboriginal people. And that was because they were used as a reliable source of fresh drinking water in areas where fresh drinking water was often really, really hard to come by. And so they were often mapped on objects like these spearheads here and through song lines. And nomadic people groups would often move from one rock hole to the next to find fresh water. The rock holes at Hilltop are actually still managed to this day by the Gawler Ranges Aboriginal Corporation. And you can see here in this photo, a rock hole that has all of this timber placed over the top and these rocks placed around the outside. And that's to prevent animals from falling in and fouling that water, as well as to limit the loss of the water to evaporation. So there's quite some serious cultural significance to these sites. And like I mentioned, they're used by a lot of animals, and part of my PhD has involved measuring this animal visitation to these rock holes. But you're probably wondering why I'm talking all about animals and I haven't mentioned any invertebrates yet. And the way that invertebrates come into the story is, as I'm sure many of us are aware, they're very, very good pool cleaners. So when you have native Australian animals that can't call out a pool cleaner to clean their pool in summer, you need someone else to provide those sort of ecosystem services. And in the desert, there are these communities of crustaceans and insects that are filter feeders. And these ecosystem services provided by these animals are pretty well documented. that all these filter feeding animals will help clean water and keep it potable for both human consumption and for consumption by vertebrates, particularly those of conservation interest. So we're interested in these invertebrate communities and how they're being impacted over time. So whether or not these invertebrate communities are changing throughout a year during the periods for which these rock holes are wet, and whether they're also changing across space. So rock holes that are further or closer to each other, do these invertebrate communities drastically change between those sites? And the technique I've been using to address this is environmental DNA, which involves, sorry, this is an image of vertebrates, but any animal that lives in a rock hole or visits that rock hole is going to leave a bit of trace DNA behind, which we can then come along and collect and then run our analyses against. So I've been doing this throughout my PhD, and I've taken 6 major field trips out to Hilltop, and on 3 of these, I've actually collected this environmental DNA samples in February, July, and October. So those are my 3 time points that I've been collecting at. So this is what it sort of looks like when you're out in the field. This is February of last year, so it was very, very hot out there, and we were running out doing these little swap missions where we would collect fresh water from the rock holes into water bottles. And the idea is that all of that loose environmental DNA is floating around in those samples, and we've collected a subset of it. We then bring that back to a lab, or sort of a field lab that we set up, and filter that through a membrane. So these membranes have a pore size of 0.2 microns, and that allows the environmental DNA, which is extracellular, to be retained on that membrane, and the wastewater that's filtered through that membrane to be discarded. And we've used these 2 systems here to do this, both of which just involve applying a vacuum to that water sample to draw it down through a membrane. Then we run a pretty standard lab pipeline. So we take those membranes, bring them back to the lab frozen, and then run an extraction across them. We quantify the DNA using qPCR and apply assays for invertebrates within the 16S and CO1 regions. And then I sent them off for sequencing through eDNA Frontiers, which allows us to generate DNA sequences, which we then blast against the database online. And that allows us to generate species IDs that should correlate to all of those loose little fragments of environmental DNA that was floating around within that rock hole. Of course, with the pandemic, my research has been slowed down somewhat, and I'm still sort of sitting at this stage. I only really reached some species IDs of confidence just last week, and this was in no small part due to assistance by Dr. Perry Beasley Hall, who sat with me all last week helping me out with the bioinformatics, which is quite new to me. So a bit of a preliminary sweep of the data and blasting it against the BLAST GenBank database. And this is for 16S, but we'll also be doing this for CO1 as well. This is 11 rock holes across those 3 time points. And these are just the top 100 features. So you can see here that we've got quite a diverse group of animals, many who you'd expect, and a few that I'm a bit less confident are actually present. And we've got about an average percentage match of 87.5%. But we're seeing lots of freshwater taxa that you expect to see in sites like this. So these are small microcrustaceans like ostracods and branchiopods, as well as chironomids, these large pea shrimp, as well as a lot of colembala, which is the dominant sequence signal at the moment, all of which I've observed in the field and have hatched out of sediments collected from these rock holes in the past. So what I'm going to be doing with this data, and this will be over the coming weeks and months, is looking at the impact of spatial variability. So how related are these communities between rock holes on an outcrop and between rock holes on neighboring outcrops of varying distances apart? So this will be comparing across between those rock holes. We'll also be looking at temporal variability. So we've got a well-studied group of rock holes on one outcrop, and we're going to be looking at how those communities change over time. So looking at the February, July, and October rainfall events and how the succession of those communities changed, and particularly expecting that they'll be quite different in February, which was the result of a summer rainfall that wasn't quite expected in the field in 2020. Finally, we're going to look at the impact of habitat variability. So I've done a bit of preliminary work in the past with community ecology, looking at the impact of habitat structure, so things like depth and plant cover on invertebrate communities in the rock holes. But what we're going to be doing now is utilizing some quite swish drone imagery that Dylan Campbell from the Unmanned Research Aircraft Facility helped me collect earlier this year, where we flew these drones over the rock holes and used photogrammetry to image each of the rock holes from a bunch of different angles, which has allowed us to generate these quite fancy three-dimensional models, which we can use to accurately characterize volume and shape, which we'll also be comparing to the invertebrate communities, which is also important for the climate change sort of vulnerability assessment. So my outcomes, and this is all going towards addressing the overarching aim of my PhD, which is what are the mitigatable threats to the role of ephemeral freshwater bodies in the Australian environment. So firstly, we've proven that eDNA can be used to monitor freshwater invertebrate communities in rock holes. And we've also improved the species inventories for the Hilltop and Nature Reserve rock holes out there in the Gawler Ranges. And in the future, I'll be testing the impact of time and space on communities using environmental DNA, and also testing the impact of habitat structure on these communities using the environmental DNA. So thank you to all of my generous funding bodies, particularly the Nature Foundation, who's allowed me access to their sites, as well as my supervisors and everyone who's collaborated and supported me on this project. Thank you. I'll take any questions now.
Thanks, Brock. If you can unshare your screen. We've already got some rapid fire questions. So Brock, do you want to read them out and answer them?
Yep, so Ari Hoffman said, to what extent do you need to be concerned about eDNA being blown in or carried in passively, which may not reflect resident species? Curious how you control for this. Yes, so we do whatever we can to control for this, and we take field blanks. So, you know, we head out into the field, open our bottles, take those water samples, and then open another bottle and, you know, decant what should be sterile water into that to look at whether or not we're getting a signal in that. And we run them through the entire process. We also do whatever we can to sort of limit contamination in the field lab as well as back in the lab back here. So it's quite a sensitive sort of lab process. Of course, I'm also elsewhere in my PhD looking specifically at vertebrate visitation through the eDNA as well. So we're actually hoping that there is that sort of carried in DNA there as well. We can test those signals. So that is part of the overall story of what we're trying to understand. But yeah, we really just do whatever we can to both control and limit contamination, but also increase our ability to characterize that contamination with those field blanks. And Daniel's asked, any idea how long eDNA lasts in these rock holes, depending on environmental conditions? Also, do you detect any eDNA from vertebrates who drink from the rock holes? Yes, so the environmental DNA is suggested to start deteriorating in as little as after 7 hours. So that's sort of as long as your water bottles last before you've got to get them filtered and frozen. Lasts a bit longer in the water column, even longer in the sediment. So that would be a future area of research to you know, try and run these same analyses against the sediment. And yes, we're specifically using 18S primers as well to look at the vertebrates who drink from the rock holes for a separate chapter of my PhD, and then going to compare that back to the invertebrate communities.
We've got one more question from Kwam, and then we'll wind it up and people can send you a direct message if they have another question.
Yep. So what taxa are being detected but not likely to be present, and why? So yes, I'm sort of, like I said, quite early in the bioinformatics stage, but there are a few things that have already, you know, sort of looked a bit dodgy to me. So I'll be sort of filtering through and looking at percentage matches, but also sort of trying to map any of that contamination, and anything that we expect not to be present but is present, we'll look at those field blanks really closely to see if it's showing up there as well. And therefore it's probably an indication of contamination at some point throughout the pipeline. Awesome, thank you for that.
Thanks, Brock.